Hi guys, this is me, this is Dr. McFarland. So I am going to try something a little bit different, and in an effort to save time, I'm going to start doing at least I think some of these online. So what I'm going to do is I am going to sort of make a recorded PowerPoint, sort of a recorded voiced over teaching presentation using the PowerPoint that I gave it, and also using some additional notes that I give you through a whiteboard drawing app. So hopefully we should be able to have a little bit less trouble with scheduling. We can meet a little less often and maybe have more substantive discussions, use more of that time for answering questions, doing practice, discussions, problems, etc. All right, so we're going to try starting off here with chapter 3, which is all about amino acids and peptides. And so this is kind of your first chapter where we actually start getting into specific biomolecules. All right, and I think amino acids are really, really interesting because not only are they the building block of proteins, but also individually and in smaller forms, the peptide forms, they do some pretty interesting things as well. All right, just a preface that my iPad does not sort of perfectly format the PowerPoint, so there will be certain cases where the pictures I put in aren't necessarily in focus. I'll try to talk around those as much as possible, and certainly we can clear up any questions or discrepancies that we have in person. All right, cool. So let's first go ahead and introduce, what amino acids are. All right, so like we said, amino acids, they're the monomers that are essentially used to build proteins. Okay? And so there are the ones that are used in proteins, there's 20 of them. There's 20 what we call essential amino acids. And whenever you link two of them together, they're joined in a peptide bond. And I'll talk more about the peptide bond in just a little bit. So let's take a look at a sample. And so you'll see me kind of periodically switching between the whiteboard and then the PowerPoint, so please bear with me. All right, so here's what an amino acid looks like. I'm going to just draw a sample, and this is one of the, first of all, I'll draw a general one, all right? So let's say we had something like this. Okay, this is an example of an amino acid. And actually, I should have, like I said, I was going to draw the general one first. So let me make an adjustment. And I'll put an R group right there. All right, this is sort of the general form of an amino acid. Let me show you the pieces. There's some lingo that you have to learn. All right, so in one of them, you're going to have a carboxy group, right? And so remember, carboxy group is going to be this. This is a carboxylic acid group right over here. And it can exist in either a protonated or a deprotonated state, right? So the term carboxy just kind of covers all general bases there. So you have a carboxy group over in this circle that is oftentimes referred to as the C terminus in an amino acid, also known as the carboxy terminus. All right, this one over here, that's an amine group, and so that's often known as the N terminus or amino terminus, right? Much like the carboxy, the nitrogen here could be in a protonated state or a deprotonated state. In a protonated state, when it's fully protonated, it's NH3, and it's got 4 bonds, so it's in positive state. When it's deprotonated, it's NH2, and it's neutral. When the carboxy terminus is protonated, it's in this carboxylic acid state, and it's neutral. If it's deprotonated, it's negative, and it looks like this, where this single oxygen has a negative charge. and then it's overall negative. The reason I highlight this is because multiple protonation states can exist. It all depends on the pH, right? So a lot of the pH stuff that you're learning right now in chapter 2 will be applicable here, and we can see how adjusting the pH will sort of tweak the protonation state of these groups and change the overall charge on the amino acid. Okay? Should I hit trash? There we go. Haven't used this whiteboard in a long time, so I'm still trying to remember all the ins and outs of it. All right, let me show you an example. Again here. So I talked about the N-terminus and C-terminus. The other thing I didn't talk about was this, the R group, all right? There's always going to be an R group, some sort of side chain that's hanging off of this carbon, the carbon that's directly adjacent to, that's sort of directly adjacent to your carboxy terminus and your amino terminus. This side chain is really what gives the amino acid its identity. It will determine specifically which type that you have, all right? There's a little, you can just remember, one of the things that you're going to have to do is you're going to have to learn all 20 structures of the amino acids, the essential amino acids. However, it's not quite as daunting as it initially looks because you're always going to have that amine group, you're always going to have that middle carbon, and you're always going to have your carboxy. That's constant, right? The only thing that really ever changes, with maybe one or two exceptions, is going to be that side chain. All right? All right, so this carbon right here is referred to as the alpha carbon. So always have an N-terminus, a C-terminus connected by an alpha carbon. And then the side chain can change depending on what type of amino acid you have. All right? So let's put an example in here of a specific one. Let's say we had a CH3. All right? If we have a CH3, that means that our amino acid is alanine. All right? Entirely decided, the identity is entirely decided by this. Okay? Depending on how long the side chain is, you can give other carbons that are in the side chain sort of letter designations, right? So this one right here, for example, if, this one right here is the alpha carbon, this one is the beta. If we were to put another one, it would be delta. I want to just sort of start building here so you can see. That would be a delta. Excuse me, gamma, sorry. My mistake. This is a gamma. Then we have the delta. And then we have the epsilon. So alanine just has beta, but they can be longer. Okay. Now one thing is, let me go back to the, whoops. That didn't work, so let me just do this. In the PowerPoint on the iPad, if you look at the third bullet point on the introduction, it didn't like my Greek letters, so it just put them as B, G, D, and E. I think it may turn out okay if you do this on a real computer, but those are not supposed to be B, G, D, and E. That is supposed to be beta, gamma, delta, and epsilon. Okay. All right, let's talk a little bit more about these. So, one thing about amino acids is that they're going to have a tetrahedral arrangement, right? And that makes sense because, the tetrahedral arrangement really is talking about sort of using the alpha carbon as sort of the centralized carbon, and it's going to have, 4 groups hanging off it, 4 different bonded groups to it. It's going to have the amino terminus, the carboxy terminus, the H, and the side chain, right? So that's corresponding to a tetrahedral arrangement. Every single amino acid has chirality. Yay, stereochemistry, woohoo! Except for glycine. The reason being that glycine looks like this. Oops, okay. Carboxy terminus, N terminus, H, and then its side chain is actually just an H, right? So if you remember from organic chemistry, can't have a stereogenic or chiral center if two of the groups are exactly the same. So this is the only achiral amino acid, at least of the 20 that we use in proteins. All right. Now, since most of them are chiral, we can actually draw the stereochemistry of that, which I know is everybody's favorite thing. However, usually in biochemistry, we don't use the same system exactly that we used to use in organic chemistry. All right. Excuse me. You might remember in organic chemistry, traditionally what we would do is, so if we had a stereogenic center, we're drawing 4 bonds from it. We're drawing 2 flat bonds that are sort of in between, they're sort of in the plane. One going away from the plane, that's going to be a dashed line, and then one coming out of the plane, that was a wedged line. And we would typically classify it as being R or S, right? So, looking at alanine, for example, we might hypothetically draw it like this. Right? That's probably what you're used to from organic. We don't really use this that often, though, in biochemistry. We use another system. Oops. All right. Instead of using the RS system, we use the LD system. All right. L stands for a term called levis, which means left. And D stands for dexter, which means right. All right. And so basically, here's what you do whenever you use the LD system. So basically what you do is you have to sort of go upon a pre-prescribed definition of how you're going to arrange things. All right, so it says to assign L or D to a molecule, look at the position of the amino group specifically in the 3D drawing. Draw the carboxy group and the side chain away from the plane, and the amino group and the H coming out of the plane. All right, so let me show you an example here with alanine. Okay, so what we do is whenever we draw these structures, we draw the centralized carbon. This is going to be, that was ugly. That's going to be your alpha carbon. All right, so you're going to take your alpha carbon, you're going to draw 2 dashed bonds on the top and the bottom, respectively. You're going to draw 2 wedged bonds on the sides. All right, so your carboxy group, let's just say we had the protonated version there, carboxy group goes up on top. Your side chain goes on the bottom, and then you have 2 groups left: the amino group, the amino terminus, and the H. Right. This, if it's in this form where the amino group is on the left-hand side, that's the L version. This would be L-alanine. Messed that up. If it's the opposite, like this, that would be D-alanine. Okay, these are called Fisher projection formulas. And there's actually more than one way to draw these Fisher projection formulas. This is just one sort of way, but it works really well whenever we draw amino acids. All right, so just by standards, it's kind of nice in one way because the carboxy group and the side chain basically already have pre-prescribed positions. You know exactly where they go, and depending on whether you have the L or the D, that just depends on where the amino group happens to be positioned. Right, and it's interesting. I mean, the position, the stereochemistry, much like we saw in organic, of amino acids makes a big difference as to how they function and what they're used for. Right. Amino acids and proteins that are found in proteins are all in the L arrangement. There are no D amino acids found in proteins. There are some D amino acids found in biochemical contexts, right? So there are some that are present, for example, in bacterial cell walls. Some are used in antibiotics, and you can find others in assorted little peptides here and there, but not any proteins. So that's kind of interesting. All right. Not too worried about this slide. This is just sort of showing how the Fisher projection was adapted to amino acids. It actually started off with a couple of aldehydes. I'm not really worried about that. All right, let's talk about the amino acids themselves. Like I said, just go ahead and tell you, you guys are going to have to learn the structures of these. So I guess we have to use the dirty M word there, memorize, right? But like I said, it's really not too bad, especially when you realize that all you have to really remember, for the most part, are the side chains, right? Depending on which book you have, will depend on how they classify them. I'm going to go with how this particular book classifies them, and it classifies them really by what their side chain is like, because, of course, that's the identifying characteristic. So if we look at the 4 different ones that we use here, all right, first we have the non-polars. Those are leucine, proline, alanine, glycine, valine, methionine, tryptophan, phenylalanine, and isoleucine. And so they are, you can see the structures. Some of this picture got cut off, unfortunately, in the on my iPad, but you should be able to see it if you're using a regular computer. So here you can see a bunch of these. You can see you can see these structures, and you can see the side chains that are highlighted in yellow. So, for example, leucine over here is going to have CH2, it's called an isopropyl group, right? Alanine has a methyl group. Glycine has an H. Sorry, didn't mean to go to glycine. Actually, never mind. This is a pretty bad picture because my iPad keeps cutting it off. My bad. When you get to this, I would say right now at this point, open your book. Open your book or open the PowerPoint on a regular computer so you can see the full pictures. So basically, if you look at all the amino acids that are listed under nonpolar, you would see that they are nonpolar, right? There's not a separation of charge there. All right? Polar ones are going to be serine, asparagine, glutamine, threonine, cysteine, and tyrosine. And remember what we talked about in chapter 1, right? Talking about polarity, or chapter 2, talking about polarity, talking about separation of charge. You're going to see a separation of charge within the side chain. I apologize if you hear my dog barking. He's being rather upset about something right now, so hopefully he'll get over it soon, right? So you can see several of them over here, or actually, whenever you open your book, because honestly, mine's kind of crapping through it right now. But threonine is a good one right here. You see, we have an OH group directly attached to a carbon. There's a distinct dipole moment between that carbon and that OH. It's polar, so it's classified as a polar amino acid. All right. Then you have the basic ones. These are the ones that actually can act as bases and will have a tendency to pick up or accept protons. All right. Those are histidine, lysine, and arginine. All right, so let me see. I don't see one on here, unfortunately. But here, tell you what, I'll draw one for you on the whiteboard just so you can see an example. Give me just a second here. Get things situated. Okay. Let's say we had this. Hold on one second. 1, 2, 3, 4. Okay. Had to count my carbons. All right, this is lysine, and this is classified as a basic amino acid. The reason it's basic is because you have this nitrogen over there, right? And we've learned in the past, from organic chemistry, you probably learned in Gen Chem before that nitrogen, having an unshared pair of electrons can accept a proton. So, and it has more of a tendency to do so than some of the other species. So, that's why lysine, it would be a basic amino acid. All right. So, all of your basic amino acids are going to have some sort of features sort of like this. All right. And I'll just stay over here on this, and I'll go ahead and draw an example of one of the acidic amino acids, and there's only a couple of them. They are aspartic acid and glutamic acid. And that makes sense, right? Those are easy to remember because, well, they have acid in their name. All right, so this one is aspartic acid. And this one has another carboxy group, much like your carboxy terminus. And we talked about the fact the carboxy terminus can be protonated, and when it's protonated, it means it's in a carboxylic acid form. Same thing over here. With aspartic acid, that O- can accept a proton. When it's in its protonated state, it exists as an acid. And of course, acids are very easily able to donate protons. So that's why this one falls into the acidic amino acid category, right? So that's one thing that might make learning these a little bit easier, is realizing which ones are in their groups and being able to look at their side chains and make that correlation. Okay. Whoops. Trying to transition back to the PowerPoint. All right. Other unique features that we can see about these as we sort of go along. All right. That's one way of classifying it. That's how your book does it. There are other ways that they can be classified and grouped besides just using those means. Some of them are sourced by, they put all the ones with alcohol groups together. in a group. Some of them use situations where they put all the aromatic ones together, or other things, right? So just sort of keep that in mind if you're using different sources. Each amino acid has two different shorthand abbreviations that it's commonly known by. One of them's a three-letter, and then one of them's a one-letter, right? And especially if you're trying to describe like a sequence of a peptide or a portion of a protein, that makes just, the shorthand notation makes it a lot easier and more efficient to describe these and write them out. So being able to recognize them when you see them is important. I'm not going to expect you to memorize these shorthands. I'll give you this table, but just be able to look at it and recognize that, see, if I have a GLY, that's a glycine. Or if I have, an N, that's an asparagine, and use that to figure out what are the amino acids in your particular sequence. All right. I'm not going to just go through all these necessarily, but I do want to highlight some particular little structures that are a little bit quirky and a little bit noteworthy. All right. So looking at the ones in the non-polar group, like we said, those are going to be the ones that don't have any dipole moments or separations of charge. Some of them have what we call aliphatic side chains, and some of them have aromatic portions. Aromatics are going to have either a benzene ring or some other aromatic ring. Aliphatic chains are just, when it says aliphatic, it just simply means not aromatic. All right. Glycine is going to be the simplest one because it's got the smallest side chain. It's got an H. All right. There's a couple of other ones that are going to be kind of their own thing because they have aromatic groups, right? Tryptophan is one, and then phenylalanine is the other one. And so I don't think I have the structures on me up here, but if you'll look in your book or at another source at the structures of both tryptophan and phenylalanine, you can see both of those have a benzene ring, and the tryptophan has an additional ring fused to your benzene that's nitrogen based. That's called an indole ring. Proline is one of the especially unique ones. This one's a little bit different from the other 19 in the respect that this actually contains a heterocyclic ring that connects the side chain with the amino terminus. All right, and I'm going to draw it out for you over here. Whoops, wrong whiteboard. Nope, not that one. Crud. Go away. All right. Sorry, Gen X struggling with technology. All right. Here's what proline looks like. Right? It's got, similar features as to what you've seen before. All right. And its N-terminus actually is an NH2 and not an NH3 in this form. And that's because you actually have a 5-membered ring that connects it with your alpha carbon. That's proline. Right? The actual side chain part of this is going to be this 3-carbon group, and that's why it's non-polar. This one is one to sort of store in the back of your minds as being special because having this ring in this position sort of gives it a unique shape. And once we get to proteins and other biomolecules, you'll see having this ring present in a particular sequence can sort of change the overall composition and shape of a protein or a peptide and make it have some unique properties. Okay, so that is something to keep in mind here. That's interesting. And then we have methionine. That's a little bit different. Methionine contains what we call a sulfide group, and that's not something that we really have gone over too much in OChem. Okay, so this is your sulfide group over there. A sulfide basically has a couple of carbons that are connected by a sulfur. Sulfur is a little bit more electronegative than carbon, but not enough to create a really big dipole moment in and of itself. So that's why it's still classified as being mostly pretty much nonpolar. This one is interesting because this one, which is methionine, is going to have some interesting interactions due to this sulfur. It can make specific types of bonds, as can another one, cysteine, which we'll talk about in just a second. They can make something called disulfide bonds. Cysteine tends to do it a little bit more than methionine, but methionine is capable of doing it sometimes. A disulfide bond is when you have a couple of sulfurs that form a sigma bond in between them. All right, I'll talk, like I said, methionine doesn't do this as much as cysteine, so I'll talk about it a little bit more in a second when I get to cysteine. But that sulfur does give methionine some unusual properties and possibilities. All right, going to the polar groups. The alcohol-containing groups, serine, threonine, and tyrosine, they all contain OHs. Asparagine and glutamine, they both contain amides, right? And again, the common thread is here we're starting to really involve nitrogen and oxygen here. That's going to create separation of charge, which means the side groups are polar. Cysteine is the other sulfur-containing one I want to highlight here. That's interesting. And so, if methionine is capable of making some disulfide bonds, cysteine is especially capable of it. Cysteine does not have a sulfide group in it, but it does have this side chain right here, and that side chain contains an SH. That is called a thiol group. And thiol groups are a little bit more reactive than sulfides. So here's where you can really see the opportunity, if I could spell it right, to make disulfide bonds. And this is interesting because when we get to proteins, we're going to talk about the varying things that sort of hold proteins into place, right? And a lot of that is the intermolecular forces that we previously talked about, like hydrogen bonds, dipole interactions, and van der Waals forces. But there are certain covalent bonds that are important in that too, and this is one of them, all right? Where do we get most of our disulfide bonds? It's going to be from cysteine that's present. Also, fun fact, and again, that goes along with the fart-based chemistry class we talked about earlier. Obviously, flatulence oftentimes does not have a pleasant smell, right? That's because a lot of the gases that are present in flatulence contain sulfur, and sulfur-based compounds oftentimes have that characteristic smell. If you smell like any commercially available thiol, for instance, it's going to smell a lot like a fart. All right, so a lot of those gases really are thiols and other related waste substances, sort of like hydrogen sulfide. Hydrogen sulfide is the one that smells sort of like rotten eggs, right? So it's kind of funny. The more foods that you consume with proteins that have disulfide bonds, those get broken down in digestion, and those form thiols, and a lot of those are gaseous, and they get released by the body. So, if you eat something that's especially rich in cysteines, then, you're more likely to have smelly farts. Important chemistry lessons for the day. There you go. Sorry for the awkward transitions here. I don't know any other way to do it. Let's see. We mentioned the acidic and the basic ones here. Again, this is gonna be interesting because when we talk about changing the pH, with most of the amino acid side chains, nothing's really going to happen to them when you change pH. It's going to be just the carboxy terminus and the amino terminus. But you can see more of it happening when you get to the acidic and the basic ones because they're more apt to either accept or donate a proton. All right. One that's really interesting to look at is called histidine. All right, let me bring up histidine. Histidine is interesting because it's the only one that's got a ring. Whoops. Come on, just draw it like your book did. Looks kind of weird, but whatever. The only one that's got a ring with a basic group in it. All right, this little fella right here. Again, sort of the awkward shape and nature of that ring can lead it to some very specific sort of stereochemical kinks and arrangements in long chains. And also the fact that this nitrogen can exist either in a protonated or deprotonated form can also affect overall charge on the molecule and attractions. So that one makes histidine a little bit unique from the others as well. All right. All right, now, those are the 20 essential amino acids, and those are the ones found commonly in proteins. They're not the only ones out there. There are other amino acids, right? We mentioned the fact that, almost all of these are going to have stereoisomers, the other forms, the D forms that aren't commonly found in proteins. But there's also a lot of them that have derivatives, right? And some of these are quite important. Oh, shoot. It cut off the picture. Okay, so at this point, make sure you have this particular picture either in your book or on your PowerPoint on your computer pulled up, and you can see some of them how they differ from each other, right? Proline, for example, they should give you the structure of proline, right? But there's also a very similar one called hydroxyproline, and the only difference between those two is that an OH group is located on one of your carbon side chains. All right, not commonly found in proteins, but it can occasionally appear in certain proteins in small amounts, and that extra OH can contribute to the properties, right? One really good example of this is collagen, right? You've probably heard of collagen. It's an important structural protein. Of course, people inject it into their lips and stuff too, right? But it's known for having, an extra bit of structural integrity, and a lot of that is because just simply including a few hydroxyprolines in there, that extra OH adds some hydrogen bonding that sort of strengthens the overall protein structure. It makes it harder to break down. Hydroxyproline is pretty common in blood plasma, right? Several other things as well. There is hydroxylysine, right? Hydroxylysine is kind of similar to hydroxyproline. It's basically a serine. Excuse me, sorry, not a serine. A lysine with an extra OH group. This is an important one that's present in a lot of receptors within the body. We're going to see a lot of proteins, will be used to fold up and make receptors. They bind other things that will either inhibit their activity or make a process more active or something like that. And different amino acid groups that are present are important in making those bindings. And so hydroxylysine is found whenever you have, commonly found whenever you need places where carbohydrates, sugars, can attach to a particular receptor. It also makes lysines more able to cross-link. And in turn, it makes the proteins that they're in more likely to cross-link. If you're not familiar what cross-linking is, basically it's a polymer thing. And this is not just with proteins, this is for all polymers. Let's say you had a polymer chain, big squiggly line, another polymer chain, big squiggly line. Maybe we have a reactive group over here. I don't know what to call it. We'll call it A. We have another reactive group over here. We'll call it B. Right? These are two individual chains. However, if, let's say, group A and group B were going to react together and form a bond, then those two chains actually become linked together. And that's where you form what we call a cross-link. And now, essentially, two big molecules have now become one because they're linked together, right? And cross-linking, the more cross-linking that you have, that can affect all sorts of things, including solubility, melting point, and also just mechanical strength. Okay, so that's pretty interesting. Another one that's not present, another amino acid that's not present even on our 20 member list. One second. Come on. There we go. Thyroxine is an important one. It's a thyroid hormone, and it's used for for regulation of all sorts of different metabolic processes, various metabolic pathways, digestion, brain health, all sorts of things. So you'll see it as an important intermediary that can either inhibit or accelerate certain processes. And yeah, it's not one of the. It's not, the amino acids are not present in the list of the 20. So there are other important biochemical amino acids that are not commonly found in proteins. All right. There's some discussion here. I'm looking at my notes, by the way. I think I'm going to save this for when we meet in person. It's pretty interesting discussion of how we start associating these amino acids with neurotransmitter activity, and then another sort of branching discussion about how we get into how these can affect depression, anxiety, and other sort of mental conditions that are caused by these neurotransmitter imbalances. So I want to save that for the regular discussion. I didn't include it in the PowerPoint, but it was in my notes. All right. All right, so let's take a look at the acid-base properties. I told you that, because we have so many different acidic and basic parts, different places where you can donate or give up a proton, C-terminus, N-terminus, and in certain side chains. These amino acids can exist in different forms, right? There's going to be at least two places where it can acquire a charge, sometimes a third place, all right? Whenever you're in doubt as to which ones will protonate or deprotonate first, check your pKa. And skipping down a little bit, here are the amino acids and the pKa values for each of their common groups that will either accept or donate protons, right? So glycine, for example, you see there's only two spots, the carboxy terminus and the N-terminus. You go a little further, though, and you see that aspartic acid, ASP, has three, right? It's got the C terminus, the N terminus, but it's also got that extra carboxy group in its side chain. So that's a third spot, and you have a third pKa, right? Tyrosine, same thing. You have a third one, except it's not going to be acidic. It's going to be basic, right? So, and the pKa values are higher once you get more basic. Remember, the pKa is basically the tendency, it gives an indication of the tendency of a group to give up a proton. All right? Generally speaking, the lower the pKa, the more of a tendency to give up a proton, or the more acidic, right? If you look at the carboxy terminus pKa's, they're all very low. That makes sense because those are carboxylic acid groups. The amino terminuses, or termini, are all much higher because NHs are considered bases, right? Aspartic acid, its side chain has a low pH. Tyrosine has a high pH. That's because those are acidic and basic side chains, respectively. All right. Now, however, regardless of the pKa, if you change the pH on your own artificially, you can manually protonate and deprotonate certain spots, right? Here's the rule of thumb, and here looking at the acid-base properties, middle slide. If you adjust the pH so that the pH is lower than the pKa, then that group will be protonated, or at least majority protonated, right? So if we go back to this chart and we look at glycine, right, the pKa of its carboxy terminus is 2.34. So if we were to lower the pH of the system containing glycine to 1, that would mean most of your carboxy termini would be protonated. They'd be in the COOH form. However, if it was above that, if it was like 5 or something like that, most of those carboxy groups would exist as COO minuses. All right. Conversely, if the pH is higher than the pKa, like I said, it exists more in the deprotonated form. Depending on the protonation states of your in your amino acids, that can change the overall charge on the amino acid, right? You could have an overall neutral molecule. You could have, a negative one, a positive one, or even, get into a negative or positive 2 in certain cases, right? So we're going to take a look at that and how that happens here. I want to introduce a bit of terminology real quick. When there's 2 charged portions individually, but the molecule is neutral overall, you can refer to it as what we call a Zwitter ion. It's pronounced Zwitter, not Zwitter, because it's a German word, right? So, let's take a look at an example. All right, let's take a look at alanine. And if you remember, alanine is the simplest amino acid. It's the one that's got, it's not the simplest. Glycine is the simplest. This is probably the second simplest one because its side chain is a methyl group. It's just a CH3, right? Since its side group is not acidic or basic, that means we're only going to have really 2 spots where we can gain or lose protons. That's going to be the C terminus and N terminus, all right? So what it does, if you, in this picture here, if you go left to right in the reaction of the equilibrium scheme, the one on the far left shows the fully protonated version. This shows you where both the NH group and also the carboxy group are fully protonated. They have all the protons they have, right? And so when your carboxy is fully protonated, it's neutral. When the nitrogen is fully protonated, it's +1. So fully protonated, that means alanine is going to have a +1 charge. All right? The first pKa, if you look at the chart for alanine, is 2.34. right? 2.34 is where the most acidic group will become deprotonated. So if you go past 2.34, what's going to happen is that carboxy group is going to be deprotonated. It's going to be in its -1 form, but it's not going to be basic enough to deprotonate. It's not going to be, it's not going to be, the solution's not going to be basic enough to deprotonate your nitrogen. So it remains in the NH3 plus 1 charge, right? So when you have a pH sort of in that range between the two pKa's, overall, your molecule tends to be neutral. It tends to be a Zwitter ion. And you see the term isoelectric. I'll talk more about that in just a minute. All right. Now, if we look at the chart, our next pKa for alanine is 9.69, right? And that's basically the pH that you have to exceed to deprotonate your amino terminus. So once we get higher than that pKa, not only is your carboxy negatively charged because it's fully deprotonated, your amino group, your amino terminus, has also been deprotonated. It's now an NH2 instead of an NH3+. So it is neutral. So at this point, -1 on the carboxy, -1 on the carboxy, neutral on the amino terminus, overall -1 charge. Okay? And you can actually represent this, and you can sort of look at it in a titration curve. Unfortunately, the picture of the titration curve here is cut off, so you can't really see what's going on. Reference the one in your book or on the full PowerPoint on the computer, and we can certainly go over this in person to talk a little bit more and see what's happening at each one of these points of the curve. All right? But this is important because changing the pH of a given medium, because it changes the overall charge on an amino acid, that's certainly going to change its behavior, right? An amino acid that's in its -1 state is certainly going to behave a little bit different than one in its neutral state, right? And this is where you're talking about attraction to various things. Other amino acids that are in the chain, incoming biomolecules that could be part of looking to bind to a receptor, right? Your receptor may not be active in a particular pH, but is active in another one because you're changing the charges on these important receptor amino acids. Several other possibilities as well, right? So it's important to be able to realize when you're going to have a negative charge, a 0 charge, +1, whatever. Okay? If you were doing this with something that had 3, I'll come back to this slide in a second. Something that had 3, right? Like aspartic acid on down. These each have 3, right? So looks like they do histidine here. And I think you can see all of them in the picture. All right. Histidine is the one with this imidazole group here, right? First pKa is 1.82. And you can see with HIS, that of course refers to the carboxy terminus, right? So below this, everything is protonated. So your carboxy is neutral, your amino terminus is +1, and this nitrogen on the imidazole ring, the one in the lower left corner of the ring, that one is protonated as well, the one that has a double bond, right? So that one's also in its positive form. So in its most protonated form, it's +2. You go above 1.82, that means the carboxy terminus will deprotonate first, right? It becomes negative. So then you have a +1 charge overall. The next one that deprotonates is 6. How do you know which one deprotonates first, the side chain or the amino terminus? Well, when in doubt, look at the chart. The lower one, the one with the lower pKa, is the one that deprotonates first. So when you get above 6, that's when your side chain nitrogen deprotonates. So now you see it's neutral. It's got the double bond on one side, a single bond on the other side, and an unshared pair of electrons. So it's neutral. Amino group is positive, carboxy is negative, overall neutral charge. And then once you get above pH of 9.17, that's when you get above the pKa of the amino group. And that's when everything is fully deprotonated, all three of those. And that's, at that point, you have a -1 charge. And the titration of that one looks like a triprotic acid, so very similar to what we saw with phosphoric. Again, since with the photo's getting cut off here, I'll go over this more in person if you need me to. All right. So you've seen a couple of places, like if you look at this third structure of histidine, you see it's labeled as an isoelectric zwitterion. And you see the same thing in the second structure of alanine. So we talked about the definition of zwitterion. We can also define something called the isoelectric point. And so basically what the isoelectric point is, that's going to be the pH that you get it to at which the molecule has absolutely no net charge, where everything is perfectly neutral. And so it's really easy to calculate. If you have something that only has 2 pKa values, like glycine, all you got to do is just add the 2 pKa's together, divide them by 2, right? So to find the isoelectric point of glycine, it would just be 2.34 plus 9.60. Let's see, I'm going to try to do this in my head. This is scary. That's 11.94, and then do 11.94 divided by 2. Oh, crap. What would that be? 5.92, I think? If it's not 5.92, please just punch it out on your calculator, but something like that, right? But point is very simple, just average the two together. That will tell you. It's a little bit more difficult if you have 3 pKa's, but still not too hard. What you do to find the isoelectric point there is you pick the 2 pKa's kind of quote-unquote surrounding neutral pH, which would be 7. So, for example, let's say we wanted to calculate isoelectric point of aspartic acid, ASP. All right? Since the 2 pHs that are sort of on either side of 7 are 9.82 and then 3.86, those would be the two that you would average together to find the isoelectric point. So 9.82 plus 3.86 divided by 2. For cysteine, which is CYS, it would be, the 2 would be 8.33 on the basic side and 1.71 on the acidic side. So 1.71 plus 8.33 divided by 2. All right. I am actually going to break this up into a couple of parts, so I'm going to stop this one for the moment. And then I'll make another video that covers the rest of this. All right, so until next one, I'll talk to you later. Bye-bye.